Good afternoon. On behalf of the Lupus Foundation of Minnesota, we want to thank everyone for joining us today for this webinar titled Updates on Research Aimed to Help Improve Treatments in African Americans. My name is Sandy Parnell, and I'm the Director of Education at the Lupus Foundation of Minnesota. I'm very pleased to introduce our presenter, Dr. Emily Gillespie. Dr. Gillespie is an Assistant Professor of Medicine and member of the Autoimmunity Program of the Center for Immunology at the University of Minnesota. She is a preceptor for students interested in careers in lupus research through the Lupus Foundation of Minnesota Student Summer Fellowship Program. Dr. Gillespie earned her PhD in 2004 from the Microbiology, Immunology, and Cancer Biology program at the University of Minnesota. Before Dr. Gillespie begins, I want to talk a bit about the functionality of the webinar system. Your webcam function should be turned off. You should be able to see the PowerPoint presentation on the screen. Your mute function is turned on. The only voices you'll hear are Dr. Gillespie's and my own. Your audio should be heard through your tablet or computer. We invite you to ask questions during the presentation. Please type in the question in the sidebar. I'll see it, no one else, and I'll ask Dr. Gillespie during or at the end of the presentation. Also, we're recording today's sessions for future audiences, and the recording will be archived on our website. At the conclusion of the webinar, a survey will arrive via your email. We hope you take a couple minutes and complete the survey because your feedback is very important as we continue to shape our webinar programming. And finally, if you'd like a certificate of attendance, please email me at sparnell@lupusmn.org. And without further delay, welcome Dr. Gillespie, and we look forward to your presentation.
All right, thank you, Sandy.
Yes.
And thanks to all of you for logging in today to participate. I'm excited to have a chance to talk to you about some of the research that we're doing in our lab. And specifically, I'll be talking about one of our research projects in which we're trying to better understand the causes of lupus in African American patients so that we can hopefully find some clues to help us find better treatments for this population. So we could go ahead to the next slide.
Okay.
All right, thanks. So I'm going to start with a visual overview of what I want to present to you today, and I'll explain each of these images more as we move through the talk. But first I want to talk about the increased frequency of lupus in African Americans and the fact that it's often more severe in these patients. And we don't really know the reasons for this, but we think there's probably several different kinds of factors that contribute to it. So these intersecting circles here sort of represent those different factors that we think might be contributing to the increased clinical burden of lupus in African Americans. And then my magnifying glass represents the fact that as a scientist, I would like to look for clues to help us understand that. And in our lab, the place where we're looking for these clues is in blood samples from lupus patients. So I'll talk a little bit about why we would look in a blood sample. And then the kind of data that we get from studying these blood samples is represented by this sort of red-green stripy square that you see there. So I'll explain more about that later as well, but this pattern actually tells us something about gene activity in the patient's blood samples. And so that we hope that what we learn from studying those patterns of gene activity is that we'll get a better understanding of why disease happens more frequently and why it tends to be more severe in African American patients, so that we can work on developing better treatments or thinking of better ways to use existing treatments for specific populations of lupus patients. Next slide, please. Thanks. All right, so why is this topic important? So this figure that we're looking at, this is part of a figure from a review article that was published earlier this year. And what it's showing us is the proportion of patients who are taking a particular medication who actually get better compared to the ones who are taking it who don't. Now, these particular medications aren't really lupus drugs, although some of them are drugs for other types of autoimmune disease. But what they are is a subset of the top 10 grossing drugs in the United States. And the blue stick figures here represent patients in whom the treatment worked, so they got better when they took it. And the red stick figures represent patients who took the same medication, but it didn't work for them. And so what we see is that for every patient in whom the treatment worked, there's anywhere from 3 to, I think it's about 24 or 25 patients who didn't get better when they took it. So what we really want to do is get smarter about how we use these medications and think about developing more targeted medications for the people who don't respond. So while again, these drugs on this slide aren't really lupus drugs, but this kind of ties back into our topic for today, because many of the original studies that set out to identify the biological processes that contribute to lupus were either conducted mainly in Caucasian lupus patients, or they were done really without regard to the race of the patients who participated. So they weren't really designed to try to answer or to try to consider the possibility that there might actually be different biological processes contributing to the disease in different subpopulations of lupus patients. And if we study this, we might be able to think of ways to have treatments that can work for a greater proportion of patients. All right, so I'm going to talk a little bit about the clinical features of lupus, and in particular, how variable they can be. So diagnosis with lupus is made using a checklist of 11 classification criteria, and this checklist includes symptoms of the disease, such as those affecting the skin or systemic manifestations, and also some of the lab tests that are used that can help with diagnosis. So any person who fulfills at least 4 out of these 11 items on the checklist is given a diagnosis with SLE. So if you think about it, there's a lot of possible combinations of items on this checklist that could get somebody diagnosed with lupus. And I think if you calculate it, it turns out to be more than 300 possible ways that these items can combine to get a diagnosis with lupus. So we were interested to try to see in a population of real lupus patients, how many different patterns could there really be? So we looked in the clinical data of a group of 300 lupus patients who participated in one of our research studies. And what we were looking for was these patterns of classification criteria, or the way that they fulfilled their checklist to get diagnosed. So there's an example shown here. This table shows each of the 11 criteria, and then it simply says yes or no for this particular patient example, whether they had that item from the checklist or not. And so if we look at the next slide, we'll see how this looked across all 300 patients from our study. I can give you a little preview. I don't see the slide up yet, but I can give you kind of a sneak peek of what we're going to see. What we found was that in these 300 patients, there were actually 175 different patterns of fulfillment of the classification criteria. So there really was a lot of variation in the ways that patients presented with and got diagnosed with lupus. And there are, if I'm remembering right, there were about 115 patterns that we saw in only a single patient. So just a unique way in which they were diagnosed with lupus. And then there were 52 clinical patterns, as I call them, that were unique to African American patients. And we didn't see those same clinical patterns in the Caucasian patients that were part of the study. So that just reinforces what we already know, that the disease is so variable from a clinical standpoint, and that there might even be different clinical features or presentations of the disease in different subpopulations of patients. So are we able to advance the slide?
Can you see it? It says African Americans are disproportionately affected, or will it go?
That is not what I'm seeing. I'm still seeing what are the clinical patterns of SLE. I don't know if that's specific to my viewing here. Sorry about that.
Okay, is this what you're seeing?
I see the slide that has what are the clinical patterns of SLE as the title of the slide.
Okay, we'll try to advance forward. Do you see the next slide?
Not yet. I know the next slide should be that black and yellow image, but I don't have that up on my screen here yet.
Well, I see the yellow and black, and I'm not quite sure what's happening, so perhaps you can keep talking, and when you see the correct slide, just give me a little shout out. Thank you.
Yeah, what I will actually do is I will open up the presentation on my own computer. I'll assume that the other attendees can see the same slide that you can.
Okay.
And for my own purposes, I'm just going to open up my own local copy so that I can see what you are all seeing. So we saw the slide with the yellow and black image, and since I wasn't seeing it, I didn't explain that the way we were showing the data was that the yellow boxes were replacing the yeses, and the black boxes were replacing the no's. So that's what we see, the yeses and no's, as we go across each row, which represents one of the patients, to see whether they had those checklist items or not. So if we can go to the African Americans are disproportionately affected slide. So down in the lower right-hand corner of that slide is that 4X. So I took that from the opening slide where I gave the visual overview. And that is to highlight the fact that the incidence rate of SLE in African American women is up to 4 times higher than it is in Caucasians. And we also know that African Americans tend to have more severe clinical manifestations and more active disease compared with Caucasian patients. African American lupus patients are also more likely to have kidney disease or lupus nephritis, as well as discoid lupus compared to white patients. So if we go to the next slide, again, we kind of have this 4X idea popping up again. Kidney disease or lupus nephritis, I abbreviated that on this slide as LN, is also up to 4 times more common in African Americans. And it's been estimated that the risk of lupus nephritis in an African American patient is 75%. It's also been shown that African American lupus patients don't respond as well to treatment, and so they often have poor outcomes despite immunosuppression. And these patients are less likely to achieve remission of their kidney disease. And in general, that predicts less favorable renal and patient survival. So overall, given all these factors, African American lupus patients tend to need more aggressive treatment. And that leads to increased risk of the toxicity that's associated with some of those treatments, like steroid treatment and immunosuppression. So let me see where we're at with the webinar here. So Sandy, are you on the slide, why is lupus more common?
Yes.
Okay. So, so we've just discussed that lupus is more common and it tends to be more severe in African Americans. And the question is why? And we don't have a good answer to that question, although we think that there are several different types of factors that probably contribute to it. Genetics is one. So as we learn more about the risk genes that contribute to risk for lupus, we think that there might be some risk genes that are more specific to one subpopulation of lupus patients than another, and that might contribute to the increased clinical burden of lupus in some patients. We also think that socioeconomic factors and environmental factors might play a role. And then from our perspective as researchers, what I'm interested in is finding some kind of objective finding to help understand this, a measurable biomarker. So that means something that I can measure in a blood sample or another kind of patient sample that will start to help explain why this happens. All right, so the next slide we're looking at is why are we looking for clues in blood samples? And so I mentioned back in that first slide that in our lab we choose to focus on primarily looking in the blood. And the reason we do that is because the blood is a rich source of immune cells. So white blood cells are the kind of cells that carry out our normal immune responses, so they help fight infection. But they also carry out autoimmune responses, and that is the sort of dangerous immune response that happens against self in a patient with an autoimmune disease like lupus. It's also relatively easy to get a blood sample from a patient compared to like a biopsy. So, you know, from a logistical standpoint, it's a good strategy for looking for biological clues about a disease. All right, so I'm going to go on to the next slide, genes encode proteins. So what kind of clues are we looking for in these blood samples? And to answer that question, we have to go back to kind of this basic principle that our genes in our DNA encode by this process the proteins, which are kind of the functional units that get the jobs done in the cells. So this picture shows our DNA, which is made up of lots of genes, and those genes can be transcribed into RNA, which is represented by that yellow squiggly, and RNA is what we refer to as the message, or the messenger RNA. And then that RNA gets translated into amino acid chains, which then fold into functional proteins, which can do the work. And in our lab, we're interested in focusing on this RNA stage, this message stage, because the RNA message level tells us something about the level of activity of all these different genes that are in our DNA. So the next slide talks about gene expression profiles. So I'm going to use this phrase a lot, and I want to explain what it means before I go any further. So every cell in our body contains the same set of genes, or the same DNA, but only a fraction of those genes are actually being used at any given moment in any particular kind of cell. So we use a bunch of different phrases or words to describe those genes that are actually being used. We might say that the gene is turned on, or that it's active, or that the gene is being expressed, and all those things refer to whether the RNA message is being made from that gene at that time and how much of it is being made. So that's gene expression, and it's the controlled pattern of gene expression that makes different kinds of cells in your body different from other types of cells. So for example, it makes a liver cell different from a muscle cell. And it also is what makes a healthy cell different from an autoimmune cell. So by looking at these patterns of gene expression, we can try to understand what it is that makes a healthy cell different from an autoimmune cell. So on the next slide, there is a kind of a visual representation of that idea. So we're talking about differential gene expression, or differences in patterns of gene expression. So this first image shows what gene expression might be like for a particular gene in a blood cell from a healthy person. So we see that the DNA from the nucleus is being made into the RNA message, which is represented by the bright green squiggly lines. And then that RNA message is getting translated into protein, which are the blue dots in that image. And then the next slide adds to this figure an example of what expression of that gene might look like in a blood cell from a lupus patient. So now we're talking about the same gene, but you see a lot more RNA message in this patient. So the gene is more active. Gene expression for this gene is higher. And so those are the sorts of patterns that we look for to give us clues as to what sort of genes might be important, because their expression is amped up in cells from a patient with lupus. So on the next slide, there's a little cartoon that explains the idea of a biological pathway, because I'm going to use this phrase a lot too, and it can mean different things at times. So I want to explain how I'll be using it. And the idea here is that we have a signaling molecule, which is represented by the purple diamond. And the signaling molecule can interact with a receptor on the surface of a cell. And so that receptor sort of catches the signaling molecule, and then it transmits a signal through the cell using these relay molecules that sort of pass it on like a baton until it ends up eliciting some kind of cellular response. And so in our case, the response that we are looking at would be these patterns of gene expression. And when we identify pathways through gene expression analysis, that can give us clues about what's happening with underlying causes for the disease. So the next slide gets to how do we do this? How do we measure gene expression? How do we figure out which genes are being turned on and when? And so we do that by focusing on the RNA message. And there are a lot of technologies that allow us to measure gene expression by measuring RNA. But it gets trickier when you want to measure RNA expression from all the genes in our genome in a single experiment. And so we're talking about measuring RNA messages from around 20,000 different genes simultaneously so that we can learn as much as possible about different pathways in a single experiment. And the next slide gives an example of how we can do this. So this is called genome-wide expression analysis, or sometimes genome-wide microarray analysis. And again, we're starting with blood samples. And so what we do is we collect a blood sample and we isolate the blood cells. And then we crack them open and isolate the RNA messages from inside them. And then we can basically give them a label and then test them out by putting them on one of these microarrays or gene chips. So the image in the middle of this slide shows a hand holding what's called a gene chip. And in this gene chip, there is a little substrate that's about the size of a postage stamp. And on that substrate are capture probes that can recognize any of the different RNA messages that you might find in the sample. And when a capture probe finds its matching RNA molecule, it'll emit a light signal from that spot. And so that's what we see in the image on the right-hand side there is those patterns of spots that are lighting up on that substrate. So we can take an image of that and turn the intensity of light into a number. And then that number relates to the amount of RNA message that was present for that gene. And by looking across the entire array, we can get those measurements for every gene in the genome, all 20,000 some measurements that we want to make all at once. So when we look at the next slide, identification of expression patterns, this gives you an idea of what it looks like after we do this. So from our blood sample and following the protocol that I described on the previous slide, we end up with a lot of data. So we're talking about 20,000 or more measurements from each patient, and we're often studying dozens or hundreds of patients in a given study. So it adds up to a lot of data that needs to be analyzed and understood. And so one way that we look at the data is called a heat map, and that's what's shown in the red and green image here on this slide. And so it's a way of looking at all this complicated data in such a way that we might be able to pick out interesting patterns and then go back and figure out if they tell us anything about the biology. And so I'll talk more in a little bit about how these heat maps work, but that gives you an idea of the way that we're going to be kind of looking at some of these data. So moving on to the next slide, just to introduce the idea of biomarkers for lupus in African American patients. And by biomarker, again, I just mean something that we can measure in a patient sample that tells us something useful about the disease. And we already know a few things. We know that there are certain autoantibodies that are found more commonly in African American patients, such as anti-RNP and anti-Smith antibodies. And I mentioned briefly the idea of genetics, that there are certain risk genes for lupus that might be more specific to one population or another. But we don't know as much about gene expression patterns in African Americans with lupus. So, for example, if we look at the recent literature, out of 11 published studies that analyzed gene expression microarrays in lupus patients, only 4 of those studies included African American patients. And of those 4, the study that had the greatest number of African American patients was 20 patients. So I think we have some gaps in our knowledge where we have the opportunity to better understand gene expression patterns in the blood cells of African American lupus patients. And that's sort of what was the motivating factor for the study that I'm going to be talking about now. So if we go to the next slide, identification of gene expression patterns in African Americans with SLE. This is the study that I'll be talking about, and that was funded for us to do by the Lupus Foundation of Minnesota. And our goal here was to take blood samples from Caucasian patients with lupus, as well as matched healthy controls, and run microarrays and understand what the expression patterns were that were different between Caucasian lupus patients and their matched control group. And then in parallel to do the same thing using blood samples from African American patients, again trying to find the expression patterns that were different in African American patients compared to their matched controls. And then in the next slide, what we do when we analyze that data is to take those two different gene lists or two different sets of expression patterns and compare them and try to figure out what's shared in the overlap between those two lists and what's unique in each population. So in a very simple way, that was really the goal of our study. So the next slide shows the methods of how we went about this. And our study included 48 African American lupus patients and 24 African American healthy controls. And then the other arm of the study had 349 Caucasian lupus patients and 32 Caucasian controls. We had a lot more patients available to the study in the Caucasian group because we were able to use data from a previous study and that was already available, already collected and generated and available for us to use in the analysis. So the novel part of the study was the ability to look at expression in the African American group. So we collected blood samples and performed the microarray analysis. And then we did a statistical analysis to find those genes that were expressed at different levels between the lupus patients and their matched controls. And looking at the next slide, there's just a table here that gives you an overview of the patient characteristics for this study. And the first row gives you an idea of what the lupus disease activity was like. So this is showing the average SLE disease activity index scores, or SLEDI. So the average SLEDI in the Caucasians was around 2, and it was around 3 in the African Americans. So similar. And in general, the average disease activity was relatively low. But then the range of activity is shown in parentheses for each group. And you can see that there were some individuals that had fairly high disease activity in both groups as well. Then the next few rows show the use of medications in the different groups. So around 30 to 40% of the patients were using immunosuppressants for their lupus. And around 50 to 60% were taking oral prednisone, and around half to 3/4 of the patients were taking Plaquenil. And as a whole, these disease activity and medication usage was fairly similar between the two patient groups, although for the oral prednisone, that little star there reminds me to say that there was a significant difference there. So the African Americans were, there was a significantly higher proportion of those patients that were taking oral prednisone compared to the Caucasian patients. And we have to keep those things in mind because medication usage has the potential to influence what we see at the level of gene expression. So that gives you an idea of the patients that were part of the study. The next slide starts to get into the results. So for the Caucasian data, again, we looked for genes that were significantly differentially expressed in the patients compared to their matched controls. And in the Caucasian group, that ended up being 735 genes. And in the comparison of African American patients to their matched controls, there were 1,280 genes in that comparison. So then the Venn diagram shows you the relationship between these gene lists. And there were 217 genes that were in common between the two groups, and that is about 12% of all the genes in these lists. And then in the Caucasian side, there were 518 genes that were specific to the gene list in the Caucasian data versus 1,064 genes that were specific to the gene list from the African American analysis. So right away what this tells us is that while there are some shared expression patterns between the two groups of patients, there's actually fairly modest overlap. So only talking about 12% or so of the expression patterns that are shared in common. And that potentially gives us the opportunity to learn something important about what's different between the lupus and these two different groups. All right, so the next slide indicates that we are going to start by sifting through the results of the shared expression profiles. So these 217 genes that were in both of our lists that potentially represent shared pathways in these two groups of patients. So what are these shared pathways? So the next slide is going to be one of these red and green heat maps. So we're going to get into this in a little bit more detail here. So this is a way of looking at the expression of these 217 genes that were in common. And the patients and the controls are shown in columns in this type of heat map. So there's a bar across the top, blue and green bar, that helps you figure out where the lupus patients are and where the healthy controls are in this figure. So those dark blue bars across the top are where the Caucasian lupus patients are, and the light blue bars are where the Caucasian healthy controls are. And then the dark green bars are where the African American lupus patients are, and the light green bars would be the African American healthy controls. So that helps you kind of navigate where the patients and controls are in this figure. The genes then are shown in rows. So every row here represents one of those 217 genes. And then we are coding gene expression on a color gradient. So green means there was low expression of that particular gene in that sample, and red would mean high expression. So we began to look at these heat maps and try to find pathways that stand out, patterns that stand out. And I should say these data have also been subjected to a type of computational analysis called clustering analysis, which attempts to put genes next to each other that share similar expression profiles. So it organizes this map. And it also tries to put people next to each other that have similar expression profiles. So it makes it easier to look for these different types of patterns within the data. So the next slide should be look a lot like the previous slide, but there's now a yellow box kind of across the top of the heat map. And that yellow box just points out one of the really prominent patterns that we saw in looking at this data set. So there's a lot of bright red within that box. That means a lot of genes that are being expressed at really high levels. And you can see that these genes are high in most of the Caucasian patients and also most of the African American patients. And when we looked at what the genes were that made up this pattern, we saw that these were genes from what we call the interferon pathway. So on the next slide, I'll explain a little bit more about what this interferon pathway is. So interferons are actually a family of proteins that help carry out antiviral immune responses. So if you are fighting a viral infection, this interferon family gets kicked into high gear and helps kick off a set of responses to help you fight off that viral infection. So interferon is actually an important part of a normal immune response. However, there's been a lot of data accumulating that implicates abnormal interferon responses in lupus. So for example, lupus patients have what we call an interferon signature, which is a set of interferon-inducible genes and proteins that are found at higher levels in lupus patients compared to controls. And the levels of these genes and proteins are correlated with lupus disease activity. So patients who have more active lupus tend to have higher levels of this interferon signature. Then when we think about the genetics of the disease, many of the lupus susceptibility genes that we know about are related to this interferon pathway. So there's evidence that abnormalities in the interferon pathway might contribute towards causing the disease. And then from a therapeutic standpoint, there are several clinical trials right now that are ongoing that are testing anti-interferon drugs in the potential treatment of lupus. So there's a pathway that we already know is implicated in this disease, and our expression data would suggest that this pathway is relevant to both of the populations of patients that we are looking at here. All right, so the next slide takes us back to that heat map of these shared genes. And now we're focusing on a different part of the heat map. So now the yellow box is enclosing a different pattern that we see. It is somewhat overlapping with the interferon pathway, but then there's a whole other group of patients that are lighting up red for this pathway that were pretty green for the interferon pathway. So there's some differences there. And when we look at what those genes were, we saw that many of them belong to the oxidative stress pathway. So when we go to the next slide, we can talk more about oxidative stress and what we know about this in SLE. So oxidative stress refers to this imbalance between the generation of reactive oxygen intermediates, or ROI, and their neutralization. And it's been suggested that there's excessive production of these reactive oxygen intermediates in lupus, as well as an impaired ability to clear them. And this might be related to development of SLE. So these reactive oxygen intermediates, they mainly come from mitochondria. And some studies have shown that T cells, which are a type of white blood cells, T cells from lupus patients have abnormal functioning of their mitochondria that sort of pushes them towards an oxidative stress state. And it's also been suggested that increased oxidative stress in lupus correlates with disease activity and with organ damage. So again, a pathway that's been previously implicated in lupus, and our expression analysis would suggest that it may be relevant both in the African American and the Caucasian population that we studied. So the next slide takes us back to the Venn diagram kind of overview of the results. And just to recap, we discussed the 217 shared genes and found a couple of dominant pathways that were represented there, the interferon pathway and the oxidative stress pathway. And so next we're going to look through those 518 genes that were specific in our Caucasian list and see what kind of clues were there. So that next slide then is going to show us the heat map showing the expression of these 518 genes that were specific to the Caucasian patients. So this is the same idea with the red and the green, but working with these array data sets, when there's so much data, there have actually been a number of different tools that have been developed to help us visualize the data and to look for those key patterns. And so I want to kind of expose you to more than one way of looking at this. So we're also going to look at these Caucasian genes in a slightly different way. So the next slide, again, represents the same data, the same expression data from the Caucasian patients, but this is using a tile plot to look at the results. And the tiles here that you see reflect different pathways or cellular functions. And the software that we used for this analysis makes some predictions about those pathways. It predicts that they're either decreased in our samples or increased depending upon the expression data that we put into the program. So the tiles that are enclosed in the red boxes here represent pathways or functions related to signaling in T cells, again, a type of white blood cells. And the fact that most of the tiles in those red boxes are blue reflects that the software predicts that there's decreased signaling in T cells in these Caucasian patients. So to talk a little bit about what that means on the next slide, I'm going to talk about what this defective T cell signaling could mean. So again, T cells are a type of white blood cell, and they carry out various jobs in the immune response. Individual T cells have a specificity, so they can recognize and attack a specific antigen. So for example, a viral or bacterial pattern that they are born to recognize. But in autoimmune disease, some T cells have a specificity for what we call self-antigens. So rather than recognizing and attacking something foreign like a virus or a bacterium, they actually recognize some part of our own body, and that's what they can attack. So it's been reported that there are defects in T cell signaling in some lupus patients, and defects in IL-2 production. IL-2 is one of the mediators of T cell function. And it's been proposed that this defective T cell signaling could be a potential therapeutic target in lupus because it could contribute to increased susceptibility to infections, which is a common problem in lupus. It could also contribute to survival of autoreactive lymphocytes. So those would be those T cells that have that specificity for self-antigens. And finally, defective T cell signaling might be involved in reduced function or number of regulatory T cells. So these are a specialized kind of T cells that could actually help prevent or limit autoimmunity. So if their function is impaired, that would be something to target in terms of potential treatments for lupus. So there's this, body of evidence that defective T cell signaling might be important in lupus and might be a therapeutic target. And the next slide is sort of, again, a reminder of what we saw in the tile plot of the Caucasian data, that there was evidence for decreased signaling in T cells based on our expression data. And if we move right along to the next slide, that will show us in the bottom half of the slide that same tile plot using the data from the African American patients. But this time inside those red boxes, instead of blue tiles, we see a lot of orange tiles or grayish tiles. So that tells us that the software is not predicting decreased signaling in T cells in the expression patterns from the African American patients. Rather, it's either not a significant change or potentially a predicted increase in signaling. So on the next slide, what that kind of means to us, what that take-home point would be, is that the Caucasian patients have this pattern of decreased expression of T cell genes, but the African American patients don't seem to show significant differences in the expression of those genes. So we perhaps should consider the possibility that some T cell directed therapies might not work the same in African Americans as they do in Caucasians, and potentially we might be able to tie some of that back to these underlying gene expression patterns. All right, so going on to the next slide, again, revisiting our Venn diagram overview here. We've just talked about some of the Caucasian-specific genes and seen that they reflect overall a decreased or defects in T cell signaling. And so lastly, we're going to take a look at the list of African American specific profiles and see what they tell us. So the next slide brings us back to our heat map way of looking at the data. And so here we're looking at the data for those 1,064 genes that were specific in the African American lists. And one of the first things we saw in looking at these data was that there were a number of interferon genes showing up here in the African American list that weren't in the Caucasian list. And that seems puzzling at first, because when we looked at the results of the shared profiles, we saw that that interferon signature was very prominent, and so we definitely felt that the interferon pathway was part of the shared group of expression profiles. But here we're seeing it's a small number, but there are a number of interferon signature genes that are showing up in the African American list. So in the next slide, we'll get into why that may be. So there is, first on that slide, there is, again, that cartoon that tells us about pathways. So the idea that there's a signaling molecule that interacts with its receptor in the cell surface, and that passes along a signal by way of these relay molecules that ultimately end up eliciting some kind of cellular response. And then we see on the right hand of the slide what that looks like specifically when we're talking about the interferon pathway. So the interferon pathway has the same kind of nuts and bolts as we saw in that cartoon, but one important difference is that there's not just one signaling molecule in this pathway. There are quite a few. So I think there are 17. I didn't actually count them just now, but I think there are 17 different subtypes that we know about right now for type 1 interferon. And all of these, they're similar, but they're different from one another, and they can all act through the interferon receptor, the same receptor in the cell surface. And then they can send signals through the relay molecules leading to their cellular response. And so I mentioned that there are some drugs in clinical trials that target the interferon pathway, and some of those drugs target the various signaling molecules, so they may target some subset or most of the different type 1 interferons themselves, while some of them target the interferon receptor or some of those relay molecules inside the cell. And I think perhaps there might be some evidence that there could be different flavors of the interferon signature in different populations of lupus patients, and that might influence the way that we think about targeting the interferon pathway therapeutically in lupus. And especially with there being so many different strategies for targeting the interferon pathway, we might learn something useful if we can better understand these different flavors of the interferon signature. All right, so there are a few more patterns that we found that were in this African American gene list. So I'm just going to kind of walk through those here on this slide. The first one was a pattern predicting an increase in something called EIF2. So that stands for eukaryotic initiation factor 2. And EIF2 is an important part of the translational machinery in the cells. So looking at that cartoon again, it's part of the machinery that takes the RNA message and helps turn it into protein. And then also part of this signature was a large group of ribosomal proteins, which also play a role in that same general process in the cell. So this EIF2 pathway has been implicated in antiviral immune responses, which is potentially relevant in lupus, and is also implicated in the production of pro-inflammatory cytokines. So these are proteins that can worsen the inflammatory response, the same type of inflammatory response that can cause damage in lupus. So the fact that we see a predicted increase in this pathway based on gene expression, specifically in African American patients, might point to the possibility of this as an avenue to pursue for therapeutics that may be particularly useful in this population. The second major pathway we saw on this slide was yet another flavor of interferon. So up until this point, I've been talking about the type 1 interferon family, which in itself has numerous different members. There's also a type 2 interferon, which is interferon gamma. And so we saw a dramatic upregulation of the interferon gamma receptor in the blood cells from African American patients, and we didn't see that in the Caucasian samples. So that suggests potentially yet another way of thinking about interferon, this time type 2 interferon, as being an important pathway in African American patients. And then finally, there was another molecule or pathway characterized by what's called IL-23. And IL-23 is gaining a lot of interest in lupus and other autoimmune diseases. This is a strong pro-inflammatory molecule again, so it tends to rev up the inflammatory response. And it's been reported to be upregulated in lupus and in some other autoimmune diseases. And it's being pursued as a potential therapeutic target in a way that makes a lot of sense with what we know about it. But interestingly, we don't find increases in this, at least in our expression patterns from the African American patients. In fact, we see it to be decreased. So this would potentially be something to consider about the use or targeting of therapeutics that target this pathway in patients with lupus. All right, so that actually wraps up what I wanted to share with you related to our research study. But I do want to show you some findings from another recent study from a different group. So this was published just a few weeks ago, I think. And this is work from Dr. Timothy Niewold's lab. Dr. Niewold is a rheumatologist and a lupus researcher at the Mayo Clinic. And he just published these results from kind of a similar idea for a study. So he was looking at gene expression in blood cells from African American and European American lupus patients. But whereas we looked at gene expression in all the white cells together, he looked at individual subtypes of white cells. So these 4 Venn diagrams on this slide represent his findings from 4 different subtypes of white blood cells. And then he similarly is showing with the Venn diagrams how much overlap he found between the African American data and the European American data. And I would just point out that similar to our findings, he found relatively little overlap in the gene list, even in these individual cell subsets, suggesting that, as we see, while there is some overlap in the gene expression profiles underlying disease in these 2 populations, there's actually far more that seems to be distinct, and that it just reinforces the idea that there may be very different biological pathways involved in lupus in different patient populations, and we should maybe consider these differences when we look at the results of clinical trials or think about new targets for therapy in different lupus populations. So just to summarize, we looked at these gene expression profiles in blood cells, and we identified a few prominent shared expression profiles that were largely dominated by the interferon pathway and the oxidative stress pathway. We also found in the Caucasian patients evidence for decreased T-cell signaling that was not seen in the African American patients. However, in the African American patients, we saw a few different things. We saw potentially different flavors of the interferon signature. We also saw an increase in the EIF2 translational machinery that could contribute towards pro-inflammatory responses. And then, interestingly, decreases in this IL-23 pathway. So overall, again, I think that what we hope to do is use this to help us understand the results of clinical trials, help us understand why sometimes these drugs don't work in certain patients, And then I have at the lower right this recycling image with some pills on there to point out that, you know, there's currently this big initiative for repositioning of existing drugs or repurposing of existing drugs. The idea being that there may be medications that are approved for use in other diseases or conditions that could potentially be useful in patients with lupus. And the question is, how do you predict out of all the other drugs that are out there, which ones might be good to test in lupus patients? And one idea is that we could use these expression profiles to help point us in the right direction. So if there are expression profiles that characterize lupus patients, those might match up with expression profiles that capture what happens in response to a particular drug that's out there for some other disease. And where those things match up, that might suggest that drug could be a good target for testing lupus. And I think our findings would suggest that we might want to think about this in terms of unique populations of patients. Rather than looking at expression patterns as a whole across all of lupus, we might want to look more at subpopulations in order to think about repurposing of other drugs that are out there based on expression profiles. All right, so I think the next slide, I just want to, before I wrap up, I want to acknowledge some of the limitations of our study and also address a couple of possible future directions for the work. So in this study, race was entirely self-reported, so we rely on the study participants to inform us about their race. We also had a much smaller sample size for the African American patients and controls compared to the Caucasians, so that could limit our ability to find those African American specific patterns. We also didn't statistically adjust for disease activity or medication use, although it seemed that these were relatively similar between our patients and future studies. I think we'd want to look more carefully at that. And then we used a pretty relaxed statistical cutoff for finding those differential expression patterns. And we did that on purpose because this was a discovery-based study. We wanted to give us the best chances of discovering those patterns that might be different. And finally, we only captured expression patterns from those circulating white blood cells. And there was also a reason for that. We thought that because they're rich in immune cells, they are probably going to be highly relevant to the disease. But it doesn't tell us about what the expression patterns might look like in the actual affected tissues and organs in lupus. So for example, if we could look in a kidney biopsy or a skin biopsy in the joint, this doesn't tell us necessarily what we would see there. Then in terms of possible follow-up work, we would want to replicate what we found in additional patients, particularly by hopefully increasing the sample size of the African American patients and controls, and see which of these observations hold up after we studied them in more patients. We would want to explore how they correlate with the different clinical features of disease. And we might want to start looking at protein levels. So RNA-based methods are great for discovery, but since proteins are sort of the level of the chain that get the job done, we may want to start looking at protein levels now that we have some clues about where to look. And then finally, we want to use this data to make some new hypotheses. So we want to start looking more carefully at the pathways that we are looking at and see if we can make some predictions about what we think is playing a role in different populations of lupus patients, and then design some studies to try to test those hypotheses. So then I think I just want to make some acknowledgments before I finish up here. So I want to acknowledge our group at the University of Minnesota, including a couple of postdoctoral researchers who have contributed significantly to this work, as well as the scientists from our lab and the study coordinators and recruiters. And then also on the right-hand side are some of our collaborators. So the collaborators listed under ABCON are those who were part of the initial effort to collect and enroll patients and collect samples to be able to do these kinds of studies. And the CLEAR investigator, Dr. Bridges, provided samples from the African American controls. And the ABCON and the CLEAR study were funded by the NIH and NIAMS. But the study that I presented here today was funded by the Lupus Foundation of Minnesota. So I want to really thank the Lupus Foundation of Minnesota for their generous support and also for the opportunity to tell you a little bit about the study today. So thank you all.
Thank you, Dr. Gillespie. We do have a couple of questions. We have about 5 minutes left.
Okay.
So let's see how many we can get through.
Sounds good.
One woman asked, given that many African Americans are not able to take high doses of dapsone, is there a drug comparable to dapsone which treats a particular type of lupus?
I am afraid I'm not going to be able to give you a good answer to that question. I think if one of my rheumatologist colleagues were on today, they could give you a very sound answer to that. But I'm not going to try to answer that question. I apologize.
Thank you. Well, you've given that person a resource to go to ask that question. Another question that popped up: what is the frequency of clinical trials, and are there enough participants? And then a follow-up question is: how might somebody become part of a clinical trial?
Okay, so in the past few years, I'm glad to say that there are a lot of clinical trials ongoing for lupus. There are clinical trials for new investigational drugs, and there are clinical trials that are designed to try to understand how combinations of existing drugs might work better together. I do hear sometimes people say, are we going to run out of lupus patients to enroll in trials, because there is a finite number of lupus patients in the country who are willing and able to participate. But I don't get the sense that that's necessarily a limiting factor yet. And then as far as how to find out about clinical trials, there is a website for lupus clinical trials, and I am embarrassed that I don't have the URL off the top of my head. But maybe, Sandy, I could work with you to kind of get that link out there. Or I think if you also possibly just Google Lupus Clinical Trials Network, that might point you in the right direction.
That's terrific. Well, that's the end of our questions, and that concludes today's webinar. Thank you so much, Dr. Gillespie, for the presentation. You've certainly raised our awareness about particular issues with women in lupus, especially African American women. We appreciate you taking the time, and thank you everybody else for joining us today for this webinar. We thank you also for your patience in the very beginning with some of our technical difficulties. Have a great day, and again, look for the evaluation form that will be coming through your email tomorrow. And this concludes the webinar. Thank you.
Thanks.